Okay, good afternoon, everybody. My name is Tourlousse. I'm a senior scientist at the National Institute of Advanced Industrial Science and Technology in Japan. So today I will be talking about our study on the comprehensive characterization of the gut microbiota of healthy Japanese to support industrialization of the human microbiome. Let me first start by thanking the organizers for giving me the opportunity to present our study. So let me start by introducing our microbiome. So the human microbiome is the collection of all microbes, mainly fungi and bacteria that live in and on the human body. The human microbiome is highly diverse, and for a typical human, healthy human has about 100 to thousands of different microbial species. And it has been estimated that each of us has around 38 trillion microbial cells, and most of them are found in the colon. This is at least as many cells as there are human cells in the body. This means that, quite amazingly, we are actually more microbial than human. And this is even more pronounced when we look at the number of genes that are encoded by the microbiome. It has been estimated that the human microbiome encodes at least 2 orders of magnitude more genes than there are genes in the human genome. So in the last decade or so, studies on the human microbiome have not only revealed the vast diversity of our gut microflora, but also showed that its composition is influenced by a wide range of factors, such as diet, exercise, medicine use, and so on. So given its vast size and diversity, it is not surprising that our gut microbiome plays a very important role in our health. And imbalances in the gut microbiome have been linked to a growing number of diseases. This includes metabolic diseases and gastrointestinal disorders such as obesity, type 2 diabetes, IBD, etc. But also diseases such as cancer and neurological disorders such as autism and depression. So, and then in the last years, we have gained a lot of understanding of the mechanisms through which microorganisms influence our health. And this has spurred a great interest from within the industry to leverage this understanding to develop new strategies for improving human health by targeting the microbiome. And globally, there are now hundreds of companies developing microbiome-based treatment options using different types of modalities, such as fecal microbial transplants, probiotics and live biotherapeutic products, small molecules, bacteriophages, etc. Interest in the microbiome was mainly driven by functional food and beverages companies. There is now a much broader range of industries that are interested in developing microbiome-based products. That includes, of course, pharmaceutical companies, but also a growing number of companies from, for example, the pharmaceutical sector and the chemical sector. So in this light, our study was initiated by the so-called Japan Microbiome Consortium. This consortium was established several years ago and includes around 40 companies from a wide range of industrial sectors. And in order to accelerate establishment of the Japanese microbiome industry, they identified 2 main goals that can be addressed through joint pre-competitive research, namely the development of microbiome community measurement standards and the development of an integrated food microbiome and health database. So this database is the main topic of my talk and is intended to be used in future studies by industry, for example, for designing intervention studies, for comparison with their patient cohorts, for discovery of microbiota or microbial species that are associated with health in order to develop new therapeutics, etc. So in order to develop this database, we analyzed the gut microflora of more than 1,000 healthy individuals from multiple regions of Japan. So by shotgun metagenomic analysis. So in total, we generated about 10 to the 13 bases worth of sequencing data. This is a very large data set and is equivalent to several thousands of human genomes. And based on this database, or based on this sequencing data, we established a comprehensive microbial genome and gene database that captures about 5,000 microbial species and on the order of 10 million different functional genes. So this database allows us to very comprehensively profile the gut microflora of the subjects in our cohort. So importantly, for all of the subjects, we also gathered extensive clinical metadata, for example, their height, weight, vital sign, blood counts, etc. In total, we have about 50 variables for each of the subjects. Each of the subjects also completed a questionnaire with around 500 questions related to their diet, exercise, sleep, stress, etc. And finally, we also performed a metabolomics analysis of blood and fecal samples, looking at about 500 different metabolites for each of the subjects. So, and by integrated analysis of these gut microflora measurements and the host variables, we can identify associations between gut microbiota and health. And then subsequently, this can lead to identification of targets that can be pursued for microbiome diagnostics and therapeutics in future studies. So as I briefly mentioned before, one of the goals of GMBC was to develop standardized methodology for shotgun metagenomic analysis of fecal samples. This is very important because different methodologies for shotgun metagenomic analysis will yield different results. So it is important to standardize those analysis techniques. And this is actually one of the main strengths of our database in the sense that we use the highly validated method to ensure that the data is reliable and can be compared with data to be generated in future studies. So all the standards that we developed are publicly available as shown in the papers on the slide. And we expect that they will contribute to strengthening the microbiome research, not only in Japan, but worldwide. So now, because we performed deep metagenome sequencing of more than 1,000 individuals in Japan, we were able to get a very detailed picture on the species diversity of the gut microflora of Japanese individuals. And this revealed hundreds of new species that had not yet previously been described or obtained in culture. So, and as I will briefly demonstrate later, several of those novel species are associated with the health of the host. So those are very important or interesting targets to be pursued in future studies. And we are now in the process of isolating most of those bacteria in our laboratory. So, and consistent with other studies over the years, we found that gut microflora of healthy Japanese is highly diverse and consists of hundreds of species. And on average, when comparing two individuals, they share only 30% of their gut microbiota. This is quite astounding considering that the human genome among different people is more than 99% identical. So the differences in our gut microbiota is much higher than the differences in our human genome. So, and as shown in the bar chart below, even at high taxonomic ranks, for example, the phylum level, we see vast differences in taxonomic abundances within our cohort. For example, the main phyla Firmicutes and Bacteroidata vary several fold among the subjects in our cohorts, which are all healthy subjects. So, and by statistical analysis, we found that the diversity both within and between the microbiome from different subjects was associated with a wide range of the host variables that we measured. This included, for example, age, where we see an increased diversity as the people age, and also gender and BMI also had an important impact of the microbiome diversity. And as a whole, for example, we found that stool consistency is one of the main drivers that of differences in microbiota between different people. So this is consistent with other studies and not only has interesting biological applications, but it also suggests that this variable should be considered, for example, in the design of future intervention studies and should be appropriately accounted for in the statistical analysis of the data sets we are generating. So based on this big data set, we are also able to discover hundreds of statistically significant associations between the abundance of specific microbes and host factors. So this includes associations that have previously been described in non-Japanese cohorts. For example, increased abundance of Prevotella species in males. And in addition, we found numerous associations that have not yet previously been described. And as highlighted, those associations often involve novel and uncultured species. And as I mentioned before, those are very interesting targets to be pursued in future studies. So as an example here, I show the results of association analysis where we looked at the gut microbiota in association with body mass index. So we found that several microbes are elevated or decreased in the people with high or low BMI. And again, those include several new types of microbes. So, and those, as a whole, this type of analysis showed that the power of our comprehensive database and extensive metadata in revealing associations between gut microbiota and health that have not yet previously been described or discovered. And this just provides numerous opportunities for developing new products such as probiotics or live biotherapeutic products for improving human health. And to conclude, by analyzing the gut microflora of more than 1,000 healthy Japanese individuals and collecting rich metadata for each of the studied subjects, we developed an in-depth knowledge database that will be very useful in future microbiome studies. So this knowledge base includes reference genome sequences for thousands of microbial species, functional and taxonomic profiles of the gut microflora across the 1,000 subjects, metabolomics data, as well as detailed health information. And based on this database, future studies can perform detailed species-resolved genome and functional gene analysis for higher resolution microbiome profiling, and also identify new species that may be targeted for cultivation. And the numerous associations that we found between the gut microbiota composition and health provides the basis for future studies, including demonstration of causation in animal studies. So, and with that, I would like to thank you for your attention. Thank you very much.